Srikanth, we're ready to start.
Yeah, should we do that? We have 24 people participating. Okay, I'll start slowly. Okay. Hi everybody, welcome to the C3.ai Digital Transformation Institute Colloquium series. So if you're in this series, I assume you know about the Digital Transformation Institute, but in case you're not familiar with it, I just wanted to give you a brief couple of minutes about what the Institute is about. So we were launched a few months ago as a consortium of 7 universities: Illinois, Berkeley, Princeton, CMU, MIT, University of Chicago, and Stanford joined more recently. And we are funded by a generous contribution from C3.ai and with cloud computing support from Microsoft. The broad aim of the Institute is to promote digital transformation of society, government, politics, using tools such as artificial intelligence, cloud computing, IoT, and so on. So as you may know, we launched a call for proposals and we selected 26 projects for funding. And so our focus during the first year is on combating the crisis that we have in our hands today, which is the COVID-19 crisis. And so the 26 awards and awardees are focused on combating this problem. And this colloquium series will at least initially focus on this particular issue, the coronavirus COVID-19, and modeling medical and healthcare issues, and how to reopen society. So all kinds of issues like that. And so that's the purpose of the colloquium series. So before I introduce the speaker for today, let me just quickly go over who the next few speakers will be after this week. So I'm not going to read all the titles. You can see them, and some of them are still a bit tentative, but the titles are tentative, but the speakers are confirmed. So the next speaker will be Alberto Sangiovanni Vincentelli from Berkeley, John Burge after that from Chicago, and then a team of Tamer Bashar, Carolyn Beck, and Becky Smith from the University of Illinois at Urbana-Champaign, Daron Asamoglu from MIT, Dimitri Bertsimas from MIT, Stefano Bertacci and his team from Berkeley, followed by Asu Ozdagler at MIT, a mathematician, Zoe Rapti from Illinois, Vince Poor from Princeton, Saurabh Amin from MIT, Sanmi Koyejo from Illinois, and finally a tag team of Sendil Mullainathan and Ziad Obermeyer from on October 8th. I mean, this is just this is not a complete list. We are expecting to have seminars every week throughout the fall semester, but I just wanted to go over the initial set of speakers. So as you can see, we have a wide variety of speakers ranging from modeling to medical imaging, like today, and how to open the economy and so on. I wanted to give you a quick idea of the format. So we'll have a 40-minute presentation followed by a 15-minute question and answer session. The attendees cannot directly ask questions, so I request you to put your questions in the Q&A box, and we will moderate and ask your questions to the speaker. And of course, you're allowed to upvote a question. So if a question has a lot of upvotes, that means it's a very popular question, and we'll pay attention to that. And we may not be able to get through all the questions, but we'll try to get answers to as many of them as possible as time permits. Okay. And if you're not the speaker, I would suggest that you turn off your video as well as microphone. So without further ado, so let me introduce our first speaker in the colloquium series. It's Professor Maryellen Geiger. We're delighted to have her as our first speaker in the colloquium series. Maryellen is a pioneer in the use of AI in medical imaging. She's a Pritzker Professor of Radiology and Medical Physics at the University of Chicago. I think around 1994 or 95, she was the first person to introduce the use of convolutional neural networks in medical imaging. And a lot of her work is not just pioneering from in an academic sense, it has been translated into practice. I'm not going to read her resume, you can read it for yourself, but she has won many, many awards. And amongst them, she's a member of the National Academy of Engineering. So today she's going to talk about her work on breast cancer imaging and then how it translates to COVID-19. Maryellen.
Thank you. I just share my screen and start. Well, thank you very much for the invitation to speak, and also thank you for the funding that we're applying to interrogate thoracic images of COVID-19. For the talk today, I will go through mainly a lot of our prior AI research in breast cancer imaging, because we're building on that in our current COVID. And I'm not moving forward. Okay. First, I need to acknowledge my grants and conflict of interest. The various grants, including the C3AI DTI grant and some other conflicts here. It's the policy of the University of Chicago that we disclose these. So the goals of my lab with AI and medical imaging is to approach a problem all the way from the lab to the clinical practice. The focus is the quantitative image analysis of images that are clinically and routinely obtained on the population. And we want to ask questions about features seen in the medical images and the biologic cancer, so that we can eventually detect and diagnose disease early, and then give the right patient the right treatment at the right time. In addition, some of the AI methods aid in improving the efficiency and the workflow in medical imaging interpretation. But before we go there, let's think about the perceptual and cognitive processes within medical image interpretation. Now, a medical image is basically a meaningless grayscale color scale pattern unless it's viewed and analyzed by an intelligent observer. That could be a radiologist, could be a computer, or could be a combination of human and computer. And the task of the human eye-brain system is to find and locate a signal, that is, what is in the image that might look like disease. Then when you focus on one potential signal in the image, is to characterize it and classify and diagnose the signal as disease or non-disease. And then do some clinical decision-making on patient management through integrated diagnostic based on the image data as well as clinical pathology and other data. Now, I'm not going to go into what is AI. I'm assuming everyone on this call, on this presentation in the audience are aware of AI. I just want to point out that AI includes many machine learning methods, including deep learning. And we have looked at these over the years, and these have been involved in our various computer-aided detection and diagnosis system, which you will learn about shortly. AI can be applied at many stages along the medical imaging chain. They can be used to optimize a detector system, or perhaps improve the image reconstruction, or even help in the workflow. But for today's talk, we're focusing on image interpretation, doing the quantitative image analysis. And in doing so, we can apply AI and develop AI for many different tasks. The work we do is task-based. One task will have different requirements from another task. In fact, I often tell my lab, think like a human when you're developing AI. What a radiologist sees in an image to make one diagnostic decision may vary compared to another, maybe therapeutic decision. So what you're looking for will vary for the human as for the computer. Listed here are various imaging tasks that one might use images, and the example I'm going to give today is in breast cancer. Risk assessment, given an image, what is the likelihood there is a future chance of cancer? Or in screening, screening implies a large amount of asymptomatic patients, and they have no other sign except what might be seen in the image. Diagnosis and prognosis is assessing something that might be found. Treatment planning, assessing treatment response, managing the patient, and even cancer discovery. Now, in looking at an image, there are many potential causes in the interpretation. There's limitations of the human eye-brain system, satisfaction of search. If radiologists someone finds a suspicious area in one part of the film of the image, maybe they might not look in any other spots. Many different causes: fatigue, workloads, distractions. And so could a computer help here? So we're almost to the research, but I want to do one more clarification. I often hear folks saying, well, they used to do AI now. I'm sorry, they used to say they did CAD and now they're doing AI, and CAD is computer-aided diagnosis. But these are loosely orthogonal. I use that loosely. Basically, AI is the algorithm one is developing that exists in a system where you input the image data to answer some specific biomedical question. However, it can be used, the output of that computer system could be used in many ways. It could be used as a second reader, where the radiologist is interpreting the image, but the output from the computer is an aid to them. That's computer-aided detection, computer-aided diagnosis. It also could be used as an aid, but as a concurrent reader. The radiologist looks at the image, and at the same time, there's an output from the computer. AI is advancing to role as a primary reader, where it looks at all the images and it might say, these 20% of images do not need to be looked at by a human. That's triaging. And that part is autonomous AI. And will we get to totally autonomous AI? Well, eventually we will have to evaluate that effect on who is the end user. Is it a radiologist? Is it another clinician? Is it the patient themselves? So I think it's very important, both in how you develop as well as how you evaluate, to know the difference between the AI and then how it ultimately will be used. And this, I didn't really specify whether it's AI to interpret the image or to also go and integrate other diagnostics into the final decision making. So let's start on the research we have been doing. So a lot of my research has been focused on breast cancer. In 2020, it's estimated there'll be almost 300,000 new cases of invasive breast cancer, and roughly 37 million mammograms will be performed each year, mainly for screening. Now, roughly half of these women will have dense breasts, so there will be additional screening images. So a lot of images. In mammography, where we use X-rays, this is the main exam for screening. Again, screening is for asymptomatic women. The energy is quite low, and what we're trying to do is differentiate the cancer from glandular tissue, and this is very difficult. This is an example. It's an old example, but it hasn't changed much. And this here is the tumor, and you can kind of see it in the prior year. It's almost gone here, but the goal of the computer is, one, to make sure you don't miss this one, but could you have found it earlier? So a lot of computer algorithms are being looked at for screening programs, and this is a localization task. Given an image, is there a suspect region in that image? And much of work is being done with screening mammograms, and this continues to today. In a screening situation, most patients will be normal. So I put here, think of the Where's Waldo book. Think of having a 1,000-page Waldo book, and he's only on 4 or 5 pages. That's screening mammography. And often it's the cancer may not be, it's not missed because it's not there, but it's not noticed. So could the computer act like a spell checker to find that? And keep in mind, the computer may not be better than a radiologist, but rather able to detect some lesions that the radiologist might miss and to work together. I know there's a lot of research going on now to make screening mammography almost an autonomous AI device, and we'll see how that goes eventually. Just for some history, this is our 1994 prototype system for computer-aided detection. This involved the detection of mass lesions and clustered microcalcifications. It's a lot bigger than what we have today, so I always like showing this. This here is the We would feed in the film. This is the storage, this is the computer, and this is the output. Here you can see, and I'm assuming you can see my arrow. This arrow is pointing to a cancer. This is a false positive, and here it missed it. This is a false negative. And that was what was used to give to the radiologist. I put this here. I was part of the team, which I believe is the first paper on using convolutional neural networks in medical imaging. This was being used to find microcalcifications on mammograms. It's very shallow, but the same concept. We did not call it a convolutional neural network. We called it a shift invariant neural network. And the way this initially worked was, here's the image. The radiologist would look at it. The radiologist is the primary reader. And once they found something, they would press the button, and then the computer output would be shown. This is actually the cancer. Here, the computer is being used as a second reader. These are false positives. And this has been translated and commercialized and has been around for a decade or so. And basically, and I give you this background so that I think we need to always learn from what's been done in the past and then learn from those developing new novel methods. It's a mixture. And basically around now, over 90% of screening mammograms have some kind of computer read. One of the issues was there are a lot of false positives. And the other issue was it was developed as a second reader, not a primary reader. And if you use it as a primary, you run into problems. Okay. And once something suspicious is found in the image, how do you know if it's cancer or not? You may do additional workup of the image. And here the goal of the computer is to help characterize and to come up with an output that's related to the question, is the lesion likely to be cancer? Again, this is often used as a second reader or a concurrent read, leaving the final decision making to the human, the radiologist. So on the left, we have a non-cancer. On the right, that's a cancer. In the middle is difficult, but that actually is a cancer. And I show this so you can see the range of presentation of lesions and the difficulty of the task. Now, in the workup, the radiologist will look at various types of modalities, trying to interrogate the lesion in order to come up with the likelihood of cancer and what should be the next step for the patient. Now, we've looked at that on two routes. And keep in mind now, the task is a characterization task. We're trying to distinguish between cancers and non-cancers. This is not a localization task. This is a characterization task. And we've looked at human engineered methods as well as convolutional neural networks. We are using clinical images. This is a clinical breast MRI image that is routinely obtained in medical centers. And with MRI, tumors have increased blood vessels and differ in their microvascular density and permeability. So if you have a cancer, if you look at the plot up on the upper right, you'll see that a cancer will uptake rapidly and then wash out. So this image at the bottom left is a pre-contrast image. You have a post-contrast in the middle. If you subtract the two, you can see the tumor pops out here. If you have benign lesions, they tend to follow the green curve. Now, this is not always, but often. And so what we do is we use this information to characterize the tumor, either visually or by the computer. So let's look at how we might use human engineered radiomics, where we're going to first segment the tumor and then extract out characteristics. We use a fuzzy C-means clustering to segment the tumor, and that's what's shown on this rotating image now. You can think of that as a virtual biopsy. And then we extract out descriptors. basically mathematical descriptors that are converted to code. We look at the size, the shape, the morphology. Are the margins of the tumor vague or sharp? We look at the texture. We look at the enhancement curve, the one I showed you in the prior ones, and how that changes over time. We then take these 3D, 4D features and merge them with some type of classifier. Many of you probably have worked with linear discriminant analysis, support vector machines, and others to yield a signature. And that signature acts as an estimate of the likelihood of malignancy. So we call this a virtual biopsy. It's automatic segmentation analysis. It's in 3D. It's relatively non-invasive, and it covers the complete tumor, and it's repeatable. So we can use this when over and over again. So what are some of the characteristics? So earlier I showed you the curve, which is shown as up here in the upper right. And while that's the kind of textbook description of the various uptakes, let's look what happens inside a tumor. So this is a breast MRI. This is the segmented tumor. I show it here in 2D, but the analysis is in 3D over time. And here are various time points. And one thing to notice is over time, for each voxel, there's a lot of different uptake curves. Some look more having rapid uptake and washout, other ones are slower. So what we did was a second round of fuzzy C-means clustering, trying to group the various type of uptakes. Also, let's say we looked at one time point where there's a lot of variation. You can look at that rate and find these most enhancing voxels. What are the voxels that are looking these most suspicious type? And those are shown here at the bottom. And then we can do 3D texture analysis. So we're taking texture analysis that you can get from a textbook, but applying it in a very specific way because we understand why the image is as it is, the physics of the acquisition, as well as the biology and medical presentation of that cancer. And we can use that as a measure of the heterogeneity of the contrast uptake. Okay, we also look at this task through using convolutional neural networks. And I'm not going to go into this in detail because this is an audience that probably knows a lot about this. We look at various types. We sometimes train from scratch, but not often because we don't have millions and millions of examples. And you think of how does a radiologist learn? A radiologist learns by a textbook, by looking at an atlas of cases and looking at typical cancers, typical non-cancers, but they also learn by going into the clinical area every day and seeing case after case after case. And that tells me we can use both of these methods together. But let's focus on the CNN method now. So we usually do transfer learning because we can get away with fewer cases. We look at fine-tuning and feature extraction methods. Today, I'm going to show you an example from feature extraction. The benefit here is we don't have to segment the lesion and we don't have to pull out features. Unfortunately, the features we do pull out are not intuitive. So here's an example where we used a VGG network and we extracted features from various levels. We used a pre-trained on the ImageNet, but we input our tumors, and then we pulled out the features that went through a conventional classifier to give us our signature again. And what I show here is a comparison. Red are cancer cases, green are non-cancer cases. And this axis on the Y shows the likelihood of being cancer as determined by the radiomic, the human engineered features, and these are from the deep network transfer learning. And what you see is in the perfect world, all the reds would be up on the upper right, all the greens would be at the lower left. But you see that sometimes the cancers are correct by the deep learning, but incorrect by the human engineered method. Other times it's the other way around. But the other thing to notice is if you draw a line like here, working together, you probably would do well. And that's what we found. We looked at various modalities. Our number of cases are much larger now. This was old from 2015. And this is the area under the ROC curve in the task of distinguishing between cancers and non-cancers. And we see that the performance is similar between the human engineered and the deep learning. And when we combined them, they all did statistically better. So that's where we are. And we ask, how do we improve the AI algorithm now in the same task? Now, people say, well, you get more cases, you do augmentation. Well, you all know about that. But I'm going to focus more on how do we change and improve by using more effective inputs or multiple input types. So one of the things is, do you help the deep net by giving more effective input versus getting more cases? And that's what we did in this study. So these three presentations are presentations used in the clinical area when a radiologist is reading. The one on the left is the central slice of the tumor, and here's the tumor, of the second post-contrast. Keep in mind, the task is saying, is this cancer or not? What is the likelihood? This is one where it's subtracted, and we can see the tumor here. And this is a maximum intensity projection, where it goes through the volume of the breast to yield this. The input to the deep net are the regions of the tumor, because that's what the radiologist is doing. They're focusing on the tumor, not on the heart. Same thing. So we pull off those regions, and that's the input to the deep net. And just as radiologists prefer this maximum intensity projection, we found also that so did the deep net. So when you don't have enough cases or a lot of cases, think what you're inputting to the network. And yes, if you have infinite amount, maybe any of these would work, but you could help it along by this, in a sense, pre-processing based on how humans do. Another example is a deep learning where we use multi-parametric MRI. And here we input different types of MRI sequences, and we looked at how we might fuse them. Do you fuse them at the different feature level, the image level, or the classifier level? But we saw overall, if you use different ways of looking at the image, you'll do better, just like you would expect from a human. And last, when we develop these, we want to eventually translate them from bench to bedside. And the road from conception and identification of the clinical problem all the way to developing the AI to completion of an FDA review to getting into the clinical involves a lot of steps. We look at potential mathematical techniques to detect or characterize lesions. We develop the methods, we create the software. We have to do it, I don't have time for this talk, but we do a lot of robust assessment developing. Validation, including reader studies, because in the end, yes, it's important to know how the computer performs, but it's also important to know is how does the user perform when they use this new AI technique? And then to translate it and make it commercial. Keep in mind, if it's endpoint, the end user, the way you have to test is with that end user. So here's an example of that kind of test. This is a very early model where we characterize those breast MRIs, and same task of distinguishing cancer from non-cancer. This is the ROC curve. This is 6 radiologists interpreting the case. This is the same 6 when they use the computer output, and you can see they improved. This is the computer alone. But what's important is how you go from this blue curve to the red curve. And that was statistically significant, and that told us the radiologist can use such an aid and to move forward. And we did, and we actually translated it. We worked with the booth in the Polsky Center at the University of Chicago. This became the first FDA cleared machine learning driven system to aid in cancer diagnosis in 2017. And we had a startup company, which is now a new company called Clarity Imaging. Now, all of this we do, keep in mind that a patient will undergo many different imaging. Patient may come in for screening. If something suspicious is found, they go for diagnostic imaging. At that point, they might get a biopsy. At that point, when they get the biopsy, we can do discovery. We can relate the images to the biopsy results. Once we do that, we may find a very useful image-based signature and translate it so that if on screening the signature shows up, that it may be a very useful biomarker, or we can use it to assess risk of recurrence. We refer to this as the virtual digital biopsy, but we would use it when an actual biopsy is not practical. One is not going to biopsy as a screening device. One is not going to biopsy repeatedly during treatment, but with imaging, you can do that. Here's an example where we looked at a single feature that we pulled off of the tumor, the most enhancing tumor volume, and we looked at how it would be to predict recurrence-free survival. Here are two cancers. This one had a recurrence, this one did not. What is different about these two? And we looked at it, but we didn't just look at all the cancers, we separated into different types of cancers. And that's shown here, where this is a very useful biomarker in distinguishing between one type of biomarker compared to another and compared to another. Did not do as well with the triple negative. So there's a lot of potential, a lot more work needs to be done here. We've also taken these features and related them to genomics. So here, for those who might be working in the genomics, this is your, these are the features that we pull off of the tumor. You can think of this as just another phenotype and do your unsupervised clustering to see if you can relate it to different characteristics of the tumor. What we found when we related these we related these features to various pathways, and I should point out, some people might say, we have 400 features from the image. Well, they really have 400 mathematical descriptors that might characterize 10 features. There's many, many texture features, and you can group them together. But what we found was the transcriptional activities of various genetic pathways were positively associated with tumor size, blurred tumor margin, and irregular tumor shape, and that microRNA expressions were associated with the tumor radiomic phenotypes of size and enhancement texture, suggesting that microRNAs may mediate the growth of tumor and the heterogeneity of angiogenesis in the tumor. Sounds like some of the features I was talking about, size, as well as texture, looking at the heterogeneity of the uptake. So now, keeping all that in mind, let's think about the parallels from breast cancer screening, diagnosis, and response to COVID-19 surveillance, diagnosis, and response to therapy. So with COVID-19, we have a need to have computational techniques to help in the early detection, in the triaging and differential diagnosis, as well in assessing prognosis, prediction, and monitoring of response. And right now, chest radiography and CT are being re-examined for their role in patient management. But keep in mind, with any imaging method, the limitations will be partially due to the qualitative interpretation of the image by the human. And so could we develop AI methods to aid in this interrogation of medical images of COVID-19 patients? And this is what the C3 AI grant is all about. We're asking, what's the role does AI? And these are our various aims. We're establishing a data set. We're developing machine intelligence for assisting in clinical decision-making. We're looking to try to investigate the explainability and visualization of COVID AI, and then also, of course, do statistical evaluation. I want to point out the research team. It's not just me. We work as a team. We have medical imaging domain experts in computational machine learning methods. That's my lab. We very much know the medical imaging domain, and we work with clinicians. That's the second one. Medical imaging domain experts in the clinical interpretation. We work with the chest radiologist, and we work with experts from Argonne in applying high performance computing. So multidisciplinary team is very important. We've been working on this since the beginning of June. Currently, we have collected 28,000 chest radiographs of over 7,000 patients, of which slightly over 1,000 are COVID-19. And people say, oh, you got a huge database, but a database is only as good as a curation. So we've now are working to curate that database and relate it to the many, many clinical values that are out on those patients to relate our AI techniques to specific clinical questions. We're building on our prior AI research in breast cancer imaging, as well as our recent work in thoracic imaging on pneumothorax detection, coronary artery disease, and emphysema. Here's an example from Jenny Crosby, where in my lab, where she looked at first identifying the type of chest radiograph, and that's very important because sometimes they're mislabeled, and then using AI to find pneumothoraces, and then doing visualization techniques to understand what is the computer really looking at. Jordan Furman, also in my lab, is looking as part of this grant at deep learning in thoracic CTs in his task, we're working with some clinical collaborators to try to use information in the CT to determine which patients should go on steroid treatment and which ones should not. And then while they're on treatment, how do they progress? We've answered the first one and gotten very promising results. And now we're analyzing it, doing a longitudinal analysis. Here's some example of heat maps that I think the explainability is a very important question. So we looked at these for pneumothorax detection. So these are pneumothoraces right here that are shown. It's when the lung falls away from the chest wall. Down here, all of these were found by the computer. But in the bottom row, what caused the computer to say this image has a pneumothorax doesn't really show where the pneumothorax is. So we're looking deeper into what do these visualization methods, this was with GradCam, how do they really show what is being interpreted by the computer? So this is early stage and we're applying it to COVID now. I do want to make a comment. Everyone says we need explainable AI. And people will say, would a radiologist or clinician trust AI if the computer output could be explained? And first is yes, they would use it if they understood where the number came from. But also, if after a while the computer is really good and they have faith in it, then they have used black boxes in the past, such as CT. Not everyone who reads a CT image knows really how that CT came about, the details of it, but they know it represents. So keep that in mind. But much research still has to be done in this field. So our goal with the C3 AI funding will be hopefully an early stage tool for surveillance and early detection of COVID-19 resurgence via monitoring chest x-rays and lung CTs. And also we hope to have an impact on assessing treatment using our virtual biopsies. So I like to end with this slide because I think this shows very important aspects. Know what you're developing, make sure it's for a specific clinical question, make sure it's task-based, and evaluate it there. But ultimately, understand how it's going to be used in the clinical arena. And because that's how the ultimate evaluation has to be done. Be careful that you're using solid images with solid truth and cautious of garbage in and garbage out. We haven't had time to go through robustness, but we dig deep when we look at what are we training our system with, what are we testing it with, how does it vary? And even that one example of the breast MRI, where if you're asking a question about the lesion, you don't need to input the entire image. You could help the AI by understanding what goes in and what comes out. So I'm going to end by thanking my lab. This is, I have outstanding graduate students, long-time outstanding folks in the research lab and various collaborators. So I can stop there. I thought I was going to be over time, but I'm not. Oops, that one.
Thank you very much, Maryellen. That was a wonderful talk. And I guess normally we would clap at the end of it, so I'll just do it. But so I guess we will open it up for question and answers. There is one question I'll start off with, I guess from Daniela Ushizima, an audience member. Her question is, why is pneumothorax relevant when the COVID-19 problem is the presence of fluid as opposed to air?
Because we're building, COVID's only been around this year, right? So we don't want to start from scratch. We're building on what we've learned. And so this talk is about my current research and how we're translating it. Just like breast cancer has nothing to do with COVID, except the various techniques that we've developed in the lab, we will also use in developing new methods for COVID. The work we've done with pneumothorax is the same way. Every time we take on a new clinical question, a new clinical abnormality, a new imaging modality, we learn something and we want to build on that. So you're correct. It has nothing to do with COVID.
Okay. Here's another question from Shuang Song. Okay, well, that's another question for you. The question is whether the recording and slides be released and where can we find it? We'll let you know. So the next question is by Robert Mark Nishikawa.
Oh, I know him.
You know him, okay. Given the urgency, he's the co-author in one of your papers, is that right?
Yes, he was at University of Chicago for a long time, so better be a good question.
Given the urgency for COVID-19 tools, how do you see the issue of clinical robustness being addressed in an efficient manner?
Well, one thing I found with the COVID is people are more willing to work with each other and everything is going in an open way to share. And I think the radiologists, I think are working together to get data sets out there as soon as possible to more and more people. I think we still have to do testing, but instead of taking years to do a clinical test, and it will of course depend on the task. For example, a patient comes in, has some symptoms, they will go under testing. We are looking at what are the characteristics of in a CT image and a chest x-ray of those who are COVID positive and those who are COVID negative, trying to understand that, because we've also learned that some asymptomatic patients who they're asymptomatic, but they test positive, but they might have lung involvement. So we should move on how that is used in the clinical arena. In there, we're trying to identify who should be going under treatment. So we need to ask that question and get rapid testing of clinicians involved, because just like COVID caused us all to go online quicker, it's going to take forever for everyone to go online, and now we're all online. I think the same thing's going to happen in getting results clinically tested. And we might show that, and I think this will actually move some of the autonomous AI of screening. Their patients will come in, might have a quick chest x-ray or CT to assess, or even more so, have the chest CT to determine what kind of treatment. And we will have to test them with radiologists, but it will be done in a more efficient way because everyone is on board now. FDA is very much involved with this. So it will move the whole field of AI in medical imaging, just like it moved the whole field of remote teaching.
Great, thank you. So the next question is from JP Gutierrez. Are you expecting to develop any new equipment or software to use the potential results from the research? How soon could that be available? I guess the question is about translating this to practice. I mean, is there research, and then how fast do you expect to translate it to software and tools?
Well, hopefully very fast, because there are initiatives that are jointly being moved along by the major imaging societies, including NIH, NIBIB, FDA. So we hope to move it along much quicker. I'm involved in the AI software end, but I do know colleagues who are working on new detector devices to move the hardware, the image acquisition.
So the next question is from Bai Hong Jin. So I noticed that the ROC curve for AI diagnosis has relatively low sensitivity at low FPR operating points, false positive rate operating points, compared to human diagnosis on slide 48. Could you comment on the reasons for that?
That's, I think slide 48 was the, oh, I know you can't see it, but I can. So basically slide 4, all right, that's the human test. That's one test. I think what you can pull from that is humans can use computer output and improve, but to start assigning overall sensitivity and specificity, you have to be careful. So when we subsequently have done this test again back in 2015, 16, and there, when a radiologist is interpreting a lesion, they're not giving a likelihood that it's cancer. And you have to ask them that in a reader study so you can do subsequently ROC analysis. Really what they're doing is saying, this lesion means that patient should go to biopsy, or no, the patient should not, maybe come in 6 months later, or nothing at all. So you have to test your sensitivity and specificity based on what is the subsequent clinical call. And I think one has to be very careful what one pulls in detail from the curve on 48, except to say that it improved the radiologist.
I have a follow-up question. So based on
Yes.
what you said, do you also then go back and figure out what happens after a biopsy and retrain your algorithm if necessary, or do you get your feedback just from the radiologist? How do you do that?
Oh, well, actually, when we train our system for diagnosis and the task of cancer versus not, we use a database of cases that we know for sure. These went on, these were biopsied and they were cancer, these were biopsied and they were benign. But then we also use benign ones that never, 2 years later, they're still benign because they never went to biopsy, but you still need that mix in your training and your testing. So the truth we use is not a radiologist truth, but more of a pathology truth.
So in that sense, are false positive rates slightly better than false negative rates? You would rather, because you also have a radiologist followed by a biopsy, whereas you don't want to miss anything. Is that how you, when you train any AI algorithm requires tuning, and then you tune the parameter to slightly lean towards FPR? Yeah.
Right. And so whether you want more false positives, whether you want higher sensitivity or higher specificity will depend on the task. So in screening, you can call everything a cancer and you'll be at 100% sensitivity, but you have many false positives. So that's not good. Now in diagnosis, if you already said this is likely to be cancer, you don't want that to be now called not to be cancer. So there you want to make sure you don't reduce your sensitivity, but it would be useful for some of the benign lesions not to have gone through the biopsy. So it always depends on the task, but then the evaluation is not just on the computer, but how the radiologists perform when they use the computer.
Okay.
This is Shankar Shastri. Maryellen, that's a wonderful talk. Your slide on the explicable AI.
Right.
Let me ask you, I think it certainly always sounds good to have explicable AI. But what is your take on situations where the AI algorithm actually tells the radiologist something that is completely surprising and something that they would not have expected? And how does one sort of prepare people for things that they just wouldn't have thought of?
Again, it's going to be task-based, I think, and that's why autonomous AI will take a while. When, for example, let's go back to the diagnosis on breast lesions. There are some cases, sometimes a radiologist will use a computer output and it agrees with what they're kind of seeing. So they just are more confident in their decision, whether it's likely to be malignant or likely to be benign. But it's the difficult cases in between. And I actually tell radiologists that if you're not sure, the computer's in the middle, it's giving you maybe a 60% likelihood or a 40%, as opposed to being at the ends. go with your gut and go with what you are assessing. You can think of a computer as a colleague. You could think of it as a resident who's still learning. This is a second opinion. But I think when the computer gives out something that is very surprising, then the radiologist needs to pause and think about why, because the radiologist is not perfect and the computer is not perfect. I mean, computers, fly airplanes and they pick movies for us, but sometimes, just like your colleague might get it wrong, your computer might get it wrong. And I think radiologists, if overall the performance is high with the computer, that's good, but I doubt if it's ever going to be perfect.
Thank you. So we have another question from Daniela Ushizima. So great talk, thanks. How are you dealing with the lack of COVID-19 CT data and respective ground truth?
Okay, so so we have a lot of chest x-rays because that's what has been done at our institution. We have collaborations with some of our colleagues in China. So we have some CTs. We have cases where the patient's been on, very sick patients put on steroids, and we've actually just submitted that one. We do have cases in Chicago now that we're collecting. They just started putting them up on the big collection site there. So that's how we're dealing with it. We just keep looking. I think one has to be careful of the quality of the images and also the truth. We verify the truth we have someone going through the clinical reports, and we also work with chest radiologists. We're very careful.
Thank you.
But you're not going to get millions of them yet. So that's why you can look at transfer learning.
So that's an interesting point, because I haven't looked at transfer learning very much, but I would think that transfer learning would work on tasks which are somewhat related, right? Given that COVID-19 is a new disease, is there an issue in using transfer learning there?
Well, yes and no. The ones we've done with the CT scan. So people do transfer learning from ImageNet, which is cats and dogs and light poles, but they use it as the earlier part, either for fine tuning or for feature extraction for medical purposes. Those are the results I've shown. And it's worked very well because the initial parts are being trained. You're not starting from scratch with weights. There's some set. But we've also started a technique that I had put into the grant, which we called Cascade. And that's what Jordan Furman is working on in the lab. It's called Cascade Transfer Learning, where first you use ImageNet to train. And we have done work with other lung diseases in the CT. And some of them are more related. Emphysema is different than COVID. Whereas some other kind of interstitial disease mimics COVID, but still it's getting a little better than ImageNet. So if you already have a trained network, that way you can do a second transfer learning, and that's what we're doing.
Okay. So there's another question from Ziyu Ye. So thank you for a wonderful talk. Have you ever encountered missing information and data sets, and if so, how do you tackle it? I guess this is a big issue in AI in general, so I would like to know what kind of information could be potentially missing in your kind of data sets, and if there are, how do you handle them?
Right, so we look at AI as being a component of a big picture. So if you say I have an image and I'm going to go all the way to the final diagnosis, you're going to run into those problems. But if you say I'm going to just handle this part and I'm going to do it really well and leave the radiologist to do the second part, you have less of that. So we've looked at methods where we have multiple ways of imaging the lesion. And then when we do that, we find that some patients might have 2 out of the 3 methods. Another one might have all 3. And while some people say you could interpolate and the word slips in my mind right now. I've lost it. I can't think of the word. Not interpolate, but you can come up with estimates of the missing data. We actually have it, if you have enough data, think of it how it's used in the clinical arena. A patient comes in, if they only have 2 imaging techniques, that's all the radiologist looks at, and they base theirs on their machine intelligence in their brain. And if the patient comes in with 3, they use it. So we use different, it's kind of like a tree then. So if the case only has 2 out of the 3, it goes through one algorithm. If it has 3 out of 3, it goes through the other. And I think you have to work that way. A lot in imaging data, we have some missing data, but not to the point of, I think they have in genomics.
Thank you. So I think if there are no more questions, I would like to thank Maryellen for her wonderful talk. It's very stimulating, and it's great to see AI being translated to this kind of societal impact. I would like to conclude by just reminding everybody that we'll have a weekly seminar series, so please join us at the same time. Next week, I guess it'll be Alberto Sangiovanni Vincentelli who'll give the talk, and he'll have a lot of exciting things to say about the uses of AI in the COVID-19 context. So please come back next week, and hope to see you then again. Thank you, and thank you, Maryellen, again. Bye.
Thank you. Bye.
Wonderful. Nice talk. Nice kickoff, guys.